Welcome back to Equity Tutors and part 2 of the circulation in mammals lessons and the final lesson in our exchange and transport series. So we left off last week having described the structure of the heart, the structure of blood vessels, why a circulatory system is important and how they are adapted to their function. So we ended up having described all of the different chambers of the heart and the major blood vessels going in and out of the heart. So following on from that, now what we want to do is to understand how the blood is moved from one chamber to the other in and out of those blood vessels. So just to recap, blood enters the heart via the vena cava into the right atrium. From there, it moves to so that's the top chamber, top left chamber, as you look at it on a diagram. From there, it moves into the right ventricle via a tricuspid valve. These are the atrioventricular valves. So these are just stopping the blood flow from moving backwards from the ventricle into the atrium. From the right ventricle, it moves out of the heart through the pulmonary artery to the lungs to collect oxygen. Oxygenated blood then returns to the heart via the pulmonary vein and enters the left atrium, so the upper right chamber. As you look at the diagram, from the left atrium, the blood moves into the left ventricle, the lower right chamber, again via an atrioventricular valve. You may call this the mitral valve. And then from the left ventricle, the blood moves out through the aorta, through the aortic valve and to the rest of the body to deliver oxygenated blood to the rest of the body. Now, I described at the beginning of last lesson the common features of a circulatory system. And one of the features of a circulatory system is a mechanism to control blood flow and move the medium, aka the blood, around the body. Basically what this means is the heart is a pump. So it's not enough that we have this pathway for the blood to move through. We actually need to physically move the blood and apply pressure for the blood to move forward and move around the body. So I'm sure, as you already know, the heart is pumping and pumps on two sides because we have a double circulatory system. So we have the right side of the heart, which is pumping blood to the lungs, and the left side of the heart pumping blood to the rest of the body. And how this pumping is regulated is actually independent of any other system in the body. So unlike any other nervous system, so for example, any other muscle moving, for instance, if you contract a muscle in your arm, that is regulated by the nervous system. The heart is completely independent of that. And the word that we use to describe that characteristic is that it's myogenic. So the heart's ability to initiate its own contraction, independent of anything else. And we refer to that as it being myogenic. So today, what we're going to do for the first half of the lesson is to just describe how that contraction, so how that force of the pump is generated. First, I just want to go over a couple of terms, two in particular, so diastole and systole. And what these mean, systole means that it's contracting and diastole means it's dilating, which means it's relaxing. So when I say that the atrium is in systole, I mean that one of the atrium is contracting and pushing blood out of the atrium into the ventricle. When I refer to ventricular systole, the same thing. I mean that the ventricle is contracting and moving blood out of the ventricle. So now let's go step by step looking at how the heart is able to initiate its own contraction. So it begins in the wall of the right atrium, so the top left chamber. And there's a region within the right atrium called the sinoatrial node. And this is essentially the pacemaker of the heart. This initiates the wave of electrical stimulation, which is going to be carried throughout the heart, all into the different chambers, which takes us through all of that blood moving through the heart. So it all starts in the right atrium via the sinoatrial node. So this initiates a wave of electrical stimulation, which causes both the atrium, so the atria, to contract at roughly the same time. So although it's located in the right atrium, the electrical stimulation initiates a wave in both atria at roughly the same time. Now this electrical stimulation is then going to move down into the ventricles, but crucially, the ventricles will not start contracting or will not enter systole until the atria have finished. And this is due to a region within the base of the atria which is unable to conduct the wave of excitation coming through. So the sinoatrial node is producing this electrical signal, is able to spread to the other side of the heart very quickly, so they contract at roughly the same time, but it doesn't spread as quickly downwards to the ventricles because you've got this region of fibers which are unable or very slowly conduct electrical signals. So we're slowing down the conduction of the signal into the ventricle so they do not contract at the same time. And this is called the septum. So the region at the base of the atria, the tissue at the base of the atria, which is unable to conduct the wave of excitation, is known as the septum. This is just a non-conducting collagenous tissue. But eventually the wave will move into the ventricles, and here it's going to the first thing that it reaches that you need to know about is the atrioventricular node. So we had the sinoatrial node in the right atrium. Now the second node we need to know about is the atrioventricular node. And this is located between the two atria, which passes the excitation onto the ventricles. If you are looking at the diagrams, which I hope you are, you can see this is labelled clearly in the diagram. So the right atrium in the top left, the non-conducting collagen tissues, and then finally just between the two atria we have the atrioventricular node. And as I always say, look at as many diagrams as you possibly can to get used to seeing different drawings, different interpretations of the anatomy, so that when these images come up in exams, you're not thrown by an image that you may not be used to. So the electrical wave eventually reaches the atrioventricular node, which is located just at the top of the ventricles, right at the bottom of the atria and in the middle of the heart, which you should be able to see on the diagrams. The electrical signal, having reached the atrioventricular node, then travels down through specialised cells which are made for conducting electricity, and we call these collection of cells the bundle of His. So these are adapted to carry the signal from the atrioventricular valve very quickly down the apex of the heart and then out into both ventricles. And the bundle of His branches off into another new term called the Purkinje fibers, and these are the part of the bundle of His which carry the signal back upwards. So you have the bundle of His which is moving down from the top to the bottom of the heart in the apex, so in the wall, the separating the right and the left regions. So that's the bundle of His coming downwards, and then after it reaches the bottom, it branches off to right and left, back upwards through the walls of the right and left ventricles, and they are referred to as the Purkinje fibers. And when the signal reaches the Purkinje fibers, this is what causes the ventricles to contract, emptying them. They're full of blood from the atria. The ventricles then contract and they move out, either up through the aorta or through the pulmonary artery. So what I want to do is to add this layer of what we've just learned, how the cardiac cycle is regulated, onto what we learned in last week's lesson in part 1, where we learned the direction of travel of the blood. Now I want to break that down step by step and think about what is happening in terms of the regulation of the right and left diastole and systole, so contraction of those chambers. So there are 3 stages to the cardiac cycle. Number 1 is cardiac diastole. Diastole, as I said earlier, means relaxed. So the heart is relaxed. Neither the atria or the ventricles are contracting. Blood isn't being pumped, and the heart is at rest. The elastic recoil of the heart. So remember we discussed last week how elastic in the blood vessels, for example, mean that they can endure changes in pressure. The heart is the same thing. So when the elastic recoils, it lowers the pressure inside the heart chambers, and what this then means is that blood can return to the heart from the vena cava and the pulmonary vein and fill the atria. So lower pressure inside the heart chambers means that any blood flow will travel down the pressure gradient into the heart via either the vena cava or the pulmonary vein and fill the right and left atria. So the right and left side of the heart are working in sync. The right atrium, left atrium are going to contract at the same time, and same for the ventricles. So as blood is entering the right atrium, it's also entering the left atrium. As it begins to fill up, pressure in the atria is going to increase. And what this means is it's going to push open the atrioventricular valves and blood flows into the ventricles. Remember, at this stage, we are still in cardiac diastole, so the atrium hasn't contracted yet. All that's happening currently is that the blood is just flowing back into the atrium, is filling up, and where that increase of pressure is happening, we've opened the atrioventricular valve and it's just passively moving into the ventricle. There's been no contraction yet. But the next step of the cardiac cycle, step 2, is atrial systole. So here is when the atrium are going to contract. And they contract because of initiation of a signal from the sinoatrial node. And when this signal is initiated, both atrium will contract. The signal then moves through a collagenous region of tissue from the sinoatrial node to the atrioventricular node, which slows down the signal. From the atrioventricular node, the signal then moves down the bundle of His and the Purkinje fibers, which will initiate step 3 of the cardiac cycle, which is ventricular systole. So I'll just explain that again. Step 1 was the heart is relaxed, blood is passively flowing into both atria because of the pressure gradient. As the atrium starts to fill up, the pressure increases, causing the atrioventricular valves to open and blood to move into the ventricles. The second step is then an initiation of the atrial systole. So the sinoatrial node generates an electrical signal, causing the atrium to contract. This then means that the remainder of the blood from the atrium will move into the ventricles. The signal generated from the sinoatrial node will also move via a collagenous region of tissue to the atrioventricular node. The atrioventricular node will send a signal to the bundle of His, which moves down the apex of the heart, up the Purkinje fibers, and leads us to step 3 of the cardiac cycle, which is ventricular systole. Now, what happens when we get contraction of the ventricles? So we know we've discussed where the blood is going to move. So the atrioventricular valves will close to prevent movement back into the atrium. Semilunar valves, so that's either the aortic valve or the pulmonary valve, will open, allowing, because of the increased pressure caused by this ventricular systole, the valves will open, allowing blood to leave out the left ventricle through the aorta or the right ventricle through the pulmonary artery. So that is a lot of information that I recommend that you write down over and over again. It's a really common exam question. The reason I split it into two is because I think there's a lot of new words to learn in one go. So it's best for you to just understand all the anatomy first before then overlaying the cardiac cycle and its regulation. You want to make sure you've got the basics understood first before you complicate things. So there's only two more small topics left on this module. And the first one, so that's hemoglobin and then tissue fluid. And I'll go over tissue fluid first because it's very quick, and then we can go into hemoglobin. First, I want you to understand what tissue fluid is. So we discussed last week blood vessels, and we know that the smallest blood vessels you get in the body are capillaries. But still, the capillaries do not have direct contact with the tissues in your body. So fat, muscle, so they need to be kept in a liquid, and that liquid is tissue fluid. So the tissue fluid will contain everything that all of your cells need to function. Things like oxygen, nutrients. So it's essentially an in-between for the circulation system of the blood and cells. So it will contain essential solutes for the cells and is also a place for exchange of waste products such as carbon dioxide. And this tissue fluid is formed due to the hydrostatic pressure when blood is pumped along the arteries into arterioles and then into capillaries. So what that means is when you're reaching the end of the path, so the blood has travelled all the way from the heart through the arteries, arterioles to the capillaries, it's reaching the end of the road. This is creating a hydrostatic pressure. The pressure builds and forces all fluid out of the blood. So basically only substances that are small enough to escape through the gap in the capillary walls. All of your smaller nutrients, components, and make up the components of the tissue fluid. Things like oxygen, glucose, fatty acid, ions, amino acids, etc. But there's also a different factor at play. So other than the hydrostatic pressure generated by the heart, so you can imagine there's a more forceful pressure, there's also osmotic pressure. So, as you can imagine, towards the end of the capillaries, where they're reaching the tissues, all of these nutrients are moving out. So, oxygen is moving out, glucose is moving out to cells which are in demand. It will then collect waste and collect water when that osmotic pressure, so where the larger elements of the blood which can't escape the ovaries are concentrated within the blood, that's going to change the water potential and mean that water moves in from the tissue fluid into the capillaries. And these are the capillaries that will then move on to become venules and veins and take waste products back to the heart. Depending on which exam board you are, you may also refer to this as oncotic pressure, but this is essentially the same as the osmotic pressure. It's all regulated by the water potential. So I'll just go over that again. Essentially, at the arteriole or artery end of the capillary, there is residual high pressure left over from the pumping of the heart. And what that means is that anything small enough to fit through the capillary wall is pushed out, forming lymph tissue. So the hydrostatic pressure is going to be larger than the oncotic or osmotic gradient generated between the capillary and the tissue fluid, and instead the hydrostatic pressure is larger, meaning that everything is pushed out. At the venous end, so the capillaries that are leading to the venules or the veins, that hydrostatic pressure is lower because we're further away from the heart, and we also have an increase in oncotic or osmotic pressure, and therefore the net movement is into the capillaries. So just to clarify, both the tissue fluid and the blood contain solutes. They have a negative water potential, but although the water potential of the tissue fluid is negative, it is less negative in comparison to the blood. Therefore, the tissue fluid is positive in comparison to the blood, and that causes water to move down the water potential gradient from the tissue fluid by osmosis back into the blood. So what is left in the tissue fluid is not pushed back into the capillaries, it actually is drained into the lymphatic system. And the lymphatic system contains the lymph fluid, similar to the tissue fluid, but will be made up of different solutes and nutrients, etc. So lymph fluid will contain less oxygen and nutrients compared to tissue fluid because its main purpose is to carry the waste products that are not going to be pushed back into the capillaries, and then they're drained in the lymph system. You may also have heard of lymph nodes, which filter out bacteria and any foreign material from the lymph nodes, which is why your lymph nodes become inflamed when you have an infection. So the final small thing that I'm going to discuss today is hemoglobin, and I've left it until the end because I'm hoping that many of you will carry on the lesson over on Patreon, because this is a topic that really requires practice in exam questions. It is usually a question which is data orientated. What I mean by that is they'll give you a graph to either draw or to analyse and to interpret the graph or the data based on your knowledge of what I'm about to teach. And this is definitely a topic which is easier to understand as you practice questions and understand how the questions are asked. So just a little bit of a reminder, what is hemoglobin? Back from Module 1, it is a water-soluble globular protein which has 2 beta polypeptide chains and 2 alpha helices. Each molecule forms a complex containing a heme group. So the heme group is the iron group, and it carries the oxygen because oxygen can bind to the heme group. And each molecule of hemoglobin can carry 4 oxygen molecules. Now, the affinity of oxygen for hemoglobin will vary. So what I mean by affinity is how easy that molecule of hemoglobin will pick up a molecule of oxygen. So the affinity of oxygen for hemoglobin varies depending on the partial pressure of oxygen. By partial pressure of oxygen, I just mean how much oxygen is around. So partial pressure of oxygen is just a measure of oxygen concentration. And the greater concentration of oxygen, so the more oxygen you have, the greater the partial pressure, and therefore the greater the affinity of hemoglobin for oxygen. So essentially, when you have low levels of oxygen, one hemoglobin molecule is much less likely to pick up a 1 molecule of oxygen, whereas when you have loads and loads of oxygen, it's picking it up very easily. And I like to refer everything back to a structure-function relationship. And the reason this is important is because in the lungs, where you have loads of oxygen compared to, for example, the stomach, you have loads and loads of oxygen. You want to pick up as much oxygen as possible in the lungs, while getting rid of as much oxygen as possible where there isn't much oxygen, if that makes sense. So where the hemoglobin is moving around the circulation system, maybe it's reaching the feet where there's low levels of oxygen. That then means that the affinity of hemoglobin for oxygen lowers and the oxygen is released in that part of the body. So that is the function for this change in affinity. In the lungs, where the hemoglobin is picking up lots of oxygen, the process is called loading. During respiration, oxygen is used up. So when we breathe in, it's immediately collected by all of the hemoglobin, and therefore the partial pressure then decreases because then we've gone back to low oxygen. Then you decrease the affinity for oxygen, for hemoglobin, and then oxygen is released in respiring tissues where it's needed. After that unloading process, the hemoglobin is then able to go back to the lungs and pick up more oxygen. So the way that we illustrate these changes in hemoglobin and partial pressure is via a graph called a dissociation curve. And this is why for this lesson I'm really encouraging you to listen to the follow-up lesson, because these questions can be difficult to answer without practicing. So these dissociation curves illustrate the changes in hemoglobin saturation as partial pressure changes. So as the amount of oxygen available changes, it's showing us how much hemoglobin is actually bound to oxygen. So the saturation of hemoglobin is affected by its affinity for oxygen. Therefore, in the case where partial pressure is high, so you have lots of oxygen, hemoglobin has a high affinity and therefore highly saturated, and vice versa. And you can see that in the example dissociation curve I have on screen. So on the y-axis, we have saturation of hemoglobin with oxygen, and the x-axis partial pressure of oxygen, or the amount of oxygen. And you can see we're increasing oxygen on the x-axis, and then the line starts to go up. You have this classic sigmoidal shape where it goes up super, super quick and then will flatten out at the top as the hemoglobin becomes saturated. So initially the curve is shallow. This is because it's hard for the first oxygen molecule to bind. So in parts of the body where the oxygen is low, we don't want the hemoglobin to pick up any more oxygen. We want to leave the oxygen available for the cells. So it's hard to pick up the first molecule. But once it is bound, the changes in the shape of the hemoglobin then make it easier for another oxygen molecule to bind and another oxygen molecule to bind until we reach 4. We refer to this process as positive cooperativity. So as one oxygen molecule binds, changes the structure of hemoglobin, makes it more likely for the next one to bind and more likely for the third one to bind. Finally, at the top of the graph, the gradient begins to flatten out, and that is because the fourth oxygen has bound and therefore no more oxygen can be picked up by the hemoglobin. The final thing you need to know about is about fetal hemoglobin. So when a woman is pregnant, she is providing oxygen to the baby. The baby is unable to breathe itself yet, although it has its own hemoglobin and its own circulatory system. The oxygen is provided for by the mother. And fetal hemoglobin is adapted for this function by having a higher affinity for oxygen compared to adult hemoglobin. Essentially what this means is the fetus is going to steal oxygen from the adult hemoglobin. So over in the Patreon lesson, we will be comparing the dissociation curves of fetal hemoglobin with adult hemoglobin to look how that may change the shape of the graph, how we interpret that, and how we can use our knowledge of hemoglobin to describe and explain those graphs. The affinity of hemoglobin for oxygen is also affected by the partial pressure or the concentration or amount of carbon dioxide. So carbon dioxide is released by respiring cells, which require oxygen. Therefore, in the presence of carbon dioxide, the affinity of hemoglobin for oxygen decreases, causing it to be released. Essentially, what this is doing is in the cells which are busy metabolically producing more carbon dioxide, is essentially a flag for the hemoglobin to say we need oxygen as we are really busy. Therefore, the carbon dioxide decreases the affinity of hemoglobin for oxygen and causes the oxygen to be released in those cells. This is known as the Bohr effect, BOHR. It does this because carbon dioxide creates a slightly acidic environment. So again, this is going to be a change in the structure of hemoglobin, changing the shape of hemoglobin, the protein, making it easier for the oxygen to be released. So that was a lot in this lesson, and I hope that you followed along. As I said, we'll be going over especially the hemoglobin affinity stuff more on the Patreon page, and especially going over questions to interpret the data. But that is all for the exchange and transport module. We've made it to the end of module 3. We will be taking a 2-week break for Easter, as I'm sure you are as well, doing lots of revision. Go back to the start and listen to all of these lessons from the beginning and to the follow-up lessons on Patreon. Those are really good in helping with exam tips and going over exam questions, breaking down how to interpret exam questions, the instruction words, how to structure your answer. And so good luck with anyone doing exams over the next couple of weeks, and I hope your revision goes well.
So thank you so much for listening. Remember, you can access additional content on our Patreon page by searching for Equity Tutors, where we have a second 30-minute lesson every week, plus monthly bonus content. You can also find us on most social media platforms. We will keep you updated on new content, and you can find us there by searching for Equity Tutors UK. Please like, share, subscribe and comment wherever you are listening, and if you're enjoying, please leave a review. Bye!
Bye!